Hello, my name is Suman Rice, and I'm chair of the Society for Reproduction and Fertility. I'm delighted to welcome you to the SRF 75th Anniversary Lecture Series. This collection of talks celebrates 75 years of the Society, its rich history, and its ongoing contributions to reproductive science. The Society was founded in 1950 as the Society for the Study of Fertility, bringing together a remarkable mix of scientists and clinicians, from reproductive physiologists to endocrinologists, pioneers of livestock breeding, and experts in human fertility. Each lecture in this series highlights the diversity of research and innovation in our field, reflecting the interdisciplinary spirit that has defined the Society from the very beginning. We hope these talks give you a sense of the Society's achievements, the work of its members, and the dynamic community that continues to advance reproductive science.
And thank you for joining this SRF Christmas lecture session. My name is Ramiro Alberio. I work at the School of Biosciences at the University of Nottingham in the United Kingdom. I'll talk today about the embryonic origin of the mammalian germline, and I will break my talk in three main sections. First of all, we will discuss what promoted germ cells are and why this is important and relevant for understanding the principles of inheritance. What is the molecular program of PGCs, what we have learned over the past few decades, and then the relationship between primordial germ cell development and the extraembryonic tissues, such as, for example, the amnion. Now, the fascination with the germline has captured the imagination of many, many scientists, particularly because of the relevance of the germline for the maintenance and continuity of the species. So August Weismann defined in the late 19th century that actually the germ cells are the repository and the source of heritable information passed between the generations. This was done for the first time at the time, and it was a very controversial discovery. But at the time, the discussion was about the role of the germline in maintaining the evolutionary path of species. And so was that Sam Butler defined this in a very pictorial way by saying that a hen is an egg's way of making another egg. So in other words, the soma, what is the body of the animal, evolved to perpetuate the germline, not the other way around. So a very egg-centric view of development and phylogeny. So moving forward many decades, about 120 years or so, we have been discussing and looking to investigate the origin of the germline in many mammalian species. Most of the work has been done primarily in the mouse as a model system because of the accessibility and the easy way to manipulate the mouse embryo. But interestingly, a few years ago, we found that the molecular program of the mouse germline differs from that of other mammals, such as, for example, domestic animal species and the human. One of the main differences is that the mouse embryo relies on SOX2 as a key gene for germ cell development, whereas in other species, the gene called SOX17 plays a crucial role in the specification of this lineage. This underlies other differences between rodents and other mammals, such as, for example, the relationship between the cells in the area highlighted here as yellow, which is the fetal part of the embryo that will give rise to the fetus, and its extraembryonic tissues. This is very different to other mammalian species. As you can see in this cartoon here, we can see that other mammals develop a flat embryonic disc, and the relationship with the extraembryonic tissues is very, very different to the rodent system. Now, what is noticeable between domestic animal species and the human is that in the human, we have an early formation of the amnion. The amnion is a layer that protects the fetus during development, and it's important during because it appears around the time of implantation. Implantation is very early in humans compared to other mammals, and this is happening around the time when gastrulation, or the first establishment of the basic germ layers, takes place in the human embryo. And therefore, the origin of the germline has always been kind of very unclear whether it had a different origin in human compared to other mammals, which gastrulate and undergo implantation much later. In fact, in humans, we have this situation where the implantation process takes place around day 9 or 10. And when the early amnion cells are forming, they are implanting into the womb. Several cells in the area of implantation around the amnion are able to produce some of the factors that are able to induce the germline efficiently. And these factors are listed here as BMPs, for example, BMP2, BMP4, and Wnt3a. These are Wnt and BMP, the two of them are important in creating a microenvironment where germ cells can appear. And so the hypothesis is that in the primate embryo, because we have early amniogenesis, some of these precursors are induced to commit to the germline as a result of the environment in which they are finding themselves. This is interesting and peculiar to humans and primates in general because they implant very early. However, it is very difficult, therefore, to separate the two events, amniogenesis from germ cell development. And whether there is a unique origin or a dual origin in the human is still highly debated. So we were interested in separating the program of the development of the germline per se from the extra-embryonic lineages, and we focus our attention to the pig as a model system where we know that amniogenesis takes place later. So this is the pink area here, the pig's amnion formation in all species. As you can see, except for primates, amniogenesis is always after the onset of gastrulation and PGC specification. So it's a good way to decouple the two events and get a good, clear picture about the germ cell specification in species that undergo late amniogenesis. So with that in mind, we investigated this across different animals. And as you can see here, we mentioned before that BMP and Wnt coordinate the specification of the germ cell lineage across all mammals. You can see here rodents, primates, and large mammals. The difference is that where the cells are specified is a bit different between the different species. And once these signals are inducing the germline program, a cascade of genes is activated within the germ cell precursors. And the two key genes, as you can see here, BMP, Wnt, and nodal, work in concert to induce the germ cell program. And this cascade of genes is triggered. The genes I would highlight as key for this talk are the ones listed, highlighted here in red. This is SOC17 and AP2 gamma, or TFAP2 gamma, as you can see, and the gene called AP2 alpha, which is a gene that has been described as critical for germ cell specification, but also for amnion formation in multiple species. So we started our investigations in the pig using 11.5 stage embryos. As you can see here, this is a top view diagram of the anterior-posterior orientation of the embryo. And if we look at the most posterior part and the cross-section, as you can see in these pictures, we can identify a cluster of cells expressing SOC17 in the most posterior part, brightly stained in the more anterior part, some cells starting to activate SOC17, followed by BLIMP1, BLIMP1 being another germ cell marker in all species. If we look a little later, about a day or so later, we can see a cluster of cells starting to form in the posterior part, increasing in size and expressing these markers SOC17 and AP2 gamma that I mentioned before. They also express BLIMP1, and they also activate nanog expression, all the key markers of the germ cell program. So we go from then a handful of cells, probably a couple of dozen cells appearing at 11.5, increasing that number to about 60 or so in the following day. If we look in more detail using confocal imaging with a top view of this embryo, these are cells that are labeled with different markers. You can see the cells labeled with SOC17, which are indicative of the hypoblast, the cells underneath the epiblast layer. But we see a domain here of cells highlighted in gray, which indicate the posterior part of the embryo, which are here in red, marking TBXT. TBXT is a marker of mesoderm. So in that area where you see highlighted in gray in this segmented image, you can see dotted cells on the border area, which are labeled in blue. These blue cells co-express brachyury or TBXT plus AP2 gamma. AP2 gamma being one of the very earliest markers of germ cells. So we identify here at the border of the epiblast with the extraembryonic tissues, which is the tissues highlighted here in light green and dark green, being the trophectoderm. At that particular border is where we identify the very first primordial germ cells in the pig embryo. They are still SOC17 negative at this point, but they're about to switch this gene on in the following few hours. These are the earliest progenitors that can be identified in this species. If we follow this for another day or so, we can see that this number, as I said before, increases from about 60 to about 120 to 200 cells in more advanced stage embryos. We wanted to know whether the increase in cells was down to DNA proliferation or recruitment of additional competent cells in the area. So to do that, we labeled the embryos with an analog of deoxyuridine, which basically allows you to quantify DNA synthesis or DNA replication. And when we look at the cluster of embryos that were labeled, you can see that SOC17 cells marks the primordial germ cells in this embryo, and none of these cells have incorporated EDU, which means that the number of germ cells when the cluster starts to form increases by recruiting new cells to the area rather than by replication of the original population. This can be depicted here, can be seen here in this histogram, where we quantify the number of cells incorporating EDU that express AP2 gamma and SOC17, a key combination for PGCs. They barely incorporate any EDU compared to somatic cells, which have high levels of EDU incorporation. So that means that as soon as the cells are specified, they will continue specifying the population of germ cells based on recruitment of neighboring cells. To gain further understanding of how this process occurs, we did single-cell transcriptomic analysis of the posterior part of the embryo. We collected basically this area is highlighted here in this little triangle here, and we performed single-cell RNA-seq of the area where the PGC cluster is located. When we do pseudotime, which is an analysis of the trajectory of the cells at different stages, we can see that the epiblast cells labeled in gray here express high levels of OCT4, which is a marker of pluripotency. And as the cells start to differentiate and undergo gastrulation, we can see a branching point here where some cells go towards the top side of this principal component graph, and some go to the lower part. The ones that go to the top part express OCT4 at this point, but they also express AP2 gamma and AP2 alpha. So see, this branch is the branch that will determine the formation of some somatic and extraembryonic tissues. The important thing is that the PGC competent area, which are the light pink cells here, are cells that express OCT4, they express brachyury, and express eomesodermin, or EOMES as we know it. And this is just before the branching towards the PGC direction, which is highlighted here by the expression of AP2 gamma and no AP2 alpha, or barely anything, and some activation of SOC17, which particularly happens in the later time point here, you can see in blue. So during this branching, we can see cells going towards the PGC program on the bottom side, and the top side going to the somatic program. To gain further understanding of this, we needed more cells. This initial study only collected about 50 cells. Now we increased that to about 400 or so cells by performing 10x single-cell transcriptome across a broader group of stages. So when we identified these cells, we were able to see that the PGCs in the pig are found before amnion formation. You can see here, this is a UMAP depicting the population of cells of the entire embryo at this point, labeled in many colors, but the important one here for us is the primordial germ cells, which appear here. And we can see that the earliest amnion progenitors start to appear around day 12.5 or so, increasing significant numbers by day 13. So the germ cells are established before the amnion in the pig without any doubt after this analysis. And what we wanted to know is what is the signaling environment in which these cells are established. So this is a little cartoon that puts two lineage trajectories. You can see the red cells here in the dotted line that go from the epiblast towards PGCs, primordial germ cells. And there are two populations of progenitors that are highlighted here, the TFAP2A precursors and PGC precursors. And as another comparative trajectory is the amnion program, highlighted here in yellow. If we look at, by doing signaling analysis, what is the type of cell signaling that these cells are exposed to? And we look at the key, three key signaling effectors, BMP, TGF-beta, and Wnt. We can see that the early TFAP2C precursors, which are the cells here highlighted in halfway the trajectory between epiblast and PGCs, these cells are highly receptive to BMP, TGF-beta, and Wnt signaling. Whereas the cells that will go towards the amnion program are highly receptive to BMP and Wnt, no TGF-beta involved in the amnion formation. So this depicts a very clear separation between the cells that are able to give rise to PGCs from those that give rise to amnion, in which nodal TGF-beta plays a critical role. And we can see this further highlighted here in this pie chart, where we can see the different populations of cells found in the embryo at this point. We can see that TGF-beta signaling network is highly active in primordial germ cells at this point of development. So having defined, therefore, that TGF-beta, BMP, and Wnt are critical, we started to look in more detail at this PGC precursor population for the genes that are expressed during that particular time. And we identified many genes related to the caudal epiblast or primitive streak markers, such as, for example, CDX4, which is upregulated in these precursors, mesodermin, and TBXD. When we look at PGC markers, we can see that this PGC precursor population activates AP2 gamma, nanos3, and SOC17. Clearly, these cells are not PGCs fully mature yet because they don't express the gene called KIT or c-KIT, which is highly expressed in PGCs themselves, but not in the PGC precursors at this time in development. So the cells are on a trajectory. They are activating sequentially a number of genes, but the key ones expressed early on are the ones listed here, AP2 gamma, nanos3, and SOC17. So we wanted to recapitulate this process using an in vitro model. Embryonic stem cells available in our lab from the pig embryo were used to try to model the primitive streak location of the cells and induction of the PGC program. For that, we took embryonic stem cells and treated them for a period of about 12 hours in the presence of activin as a representative of the TGF-beta program, Wnt signaling induced by chiron, a small molecule, and BMP4. When we applied this for 12 hours, we induced moderate amounts of brachyury in all these cells. We maintained the levels of NANOC, and we induced moderate amounts of mesodermin. So this is an indication that the cells are entering or engaging into the primitive streak program. We follow this treatment by inducing the PGC program using BMP and a combination of activin, chiron, and BMP for another day and a half. And as you can see, many cells turned yellow, which is the combination of AP2 gamma and SOC17, indicative of the specification of these cells in vitro. With this in mind, we decided to address a question that was recently addressed in the literature, which shows that in the human, in primates, AP2 alpha is a key determinant of the human germline. So if you do a knockout for AP2 alpha in humans, the germ cells are basically completely depleted or they die soon after. Because the origin appears to be different or at least of multiple origins in humans, dual origin in humans, but not in pigs, we decided to deplete AP2 alpha from our stem cells and establish whether they could make PGCs. And using CRISPR gene editing, we did this in our AP2 alpha cells, were generated knockout cells, and we induced the same induction protocol. And as you can see in these images, SOC17 and AP2 gamma dual positive cells were detected in similar numbers compared to the wild type cells, indicating that AP2 alpha is not required in PGC specification in the pig species. Now, moving on to some other important aspects of PGC development relate to the epigenetic reprogramming of the germline. This is a fundamental event that takes place only in germ cells, not in other somatic cells, and which consists in the erasure of epigenetic marks early on in development to ensure that there are no epimutations that are transferred to the next generation. So we decided to investigate this in our cells in vivo, in the pig embryo, and we looked at a series of markers, as you can see here in these sections of embryos, which show PGCs at different points in their migration or location within the cluster of PGCs. So you can see that DNMT3A, which is a marker of de novo methyltransferase activity, is reduced in PGCs, which is marked with OCT4 compared to the neighboring cells. We also see UHRF1 very much depleted in the cluster of PGCs identified in this embryo. We also see a reduction of H3K9 dimethylation and an increase in TET1. This was all confirmed by the RNA-seq data that we showed you before, where we see a reduction in the PGC precursors of UHRF1, DHMT2, and DNMT3B, indicating that around the time of PGC specification, the epigenetic reprogramming is already on its way. This is very, very early, really, considering what's been described in other species, like, for example, in rodents. To learn a bit more about this, we looked and measured in more detail using mass spectrometry the content of 5-hydroxymethylcytosine in these cluster cells. The cells are here labeled in green, are 5-hydroxymethylcytosine stained cells. These are the PGCs labeled with OCT4. One can see here in the entire cross-section of this embryo, there is a very big enrichment for 5-HMC in the PGCs. If we measure this, as I said, by mass spectrometry, we can see a significant increase in TET1, also in 5-HMC expression labeling, which indicates a TET1 activity. And this is an indication of active DNA demethylation. If we look at the reactivation of the X chromosome, which happens in females at this time of development, you can see here a PGC marked with SOC17. We can see that this cell here does not have this particular dot that is present in all the somatic cells at this time of development, which indicates the inactive X. And so when we sequence these cells, we can see a 20% increase in the expression of genes in the PGCs at day 14 compared to the somatic cells, indicating a reactivation of the X chromosome before the PGCs initiate their migration. We decided to also look at the resetting of the DNA methylation of the PGCs in gonadal cells, and we could see that only 1% of cells retain DNA methylation in the gonads in the pig. When we look specifically at what kind of regions these were, there were regions that were rich in transposable elements. And when we look at the type of transposable elements, we saw that the majority of these regions retaining methylation are evolutionary young transposable element regions. When we look specifically what kind of genes and synthetic regions they exist between human and pig, we found about 21% overlap between human and pig synthetic regions. And about 250 genes or so were found in common between these two species, which allowed us to do a GWAS analysis to identify genes that are common between pigs and humans that might escape epigenetic reprogramming. They're called escapees. These are genes that are passed on between generations that do not undergo erasure of DNA methylation. This is quite significant because the genes that we found associated with this loci are genes related to metabolic disorders and neurological disorders, suggesting that perhaps there is a link between epigenetic inheritance of germline, epigenetic inheritance related to the lack of erasure of epigenetic marks in the mammalian germline. So with this, I'd like to give you a brief summary of what I presented to you today, where I showed you that the earliest PGCs in the pig originate from posterior epiblast cells that are identified by the expression of AP2 alpha, TBXD, SOC17, and NANOS3. I also show you that the amnion progenitors emerge after PGC specification in the pig species, and the amnion marker AP2 alpha is not required for pig PGC specification. I also showed you that pig primordial germ cells initiate epigenetic reprogramming before their migration to the gonadal ridges, and that pig and human escapees could act as suitable proxies for understanding the role of transgenerational inheritance of specific epimutations. So With this, I'd like to conclude my talk by acknowledging my colleagues involved in this study, the people listed here in red, Daniel Gosinski, Doris Klisch, Ahmed Ramzi, and Luke Simpson, who have contributed specifically to this project, and my collaborators in Cambridge, Azim Surani, and in Okazaki University, Toshi Kobayashi. And of course, I'm grateful to my funders, BBSRC, for the generous support in this program of work, and our current funders, Wellcome Trust and Gates Foundation, which are helping us continue this work going forward. Thank you for your time, and I hope you enjoyed this talk.